So yeah, as Anjana said, I'm going to talk about manipulating memories. And I'm from the Tonegawa lab. The Tonegawa lab is the Pickower Institute of Learning and Memory, and we study basically how memory is encoded in the brain, how it changes, what happens to it over time, how it changes when we recall things, how it changes when we forget things, and sort of the cellular and behavioral correlates of memory. And so I always begin my talk by asking this question, like, what is memory? And a lot of people have asked this question before me. And one of the first was Plato from Athens, who was a big philosopher back in the day. And one of the newest people to have asked this question is Richard Seaman, who was also a psychologist, physiologist, sort of a neuropsychophysiologist in 1859 in Germany. Of course, they are not the only people to have asked this question. There have been more people before and after, but these are important, and I'll tell you why. Why is memory important? Memory is important because we have to learn. We have to learn about the world around us, and it's important to remember what we learn in that instance so that we can build on knowledge that we know. To socialize, it's, I think, one of the most important things as human beings, as social animals. It's important to remember who we're socializing with, to link our past and future. So to learn and to socialize, this is sort of the core concept. You need to link what you know in the past, what you remember from the past, to how you predict your future is going to be. So memory is sort of this very core phenomenon that subserves or underlies a lot of very sort of key behaviors in general. So one of the things that we do in our lab is to mark memories, and what we do is use a couple of different proteins and a couple of different tools for this. And these two proteins obviously are not very they don't elicit any memory, or they don't elicit any recognition, but they're actually very key important proteins for us in the lab. And the top here is GFP, which I know a lot of people will know. And GFP is basically green fluorescent protein that's isolated from a jellyfish called Achoria victoria. And the bottom one right there is Chandlerodopsin, which is isolated from a really small algae. It's called Caenorhabditis, and sorry, not Caenorhabditis, Chlamydomonas, Chlamydomonas reinhardti. And so these two proteins together are what basically make our lab tick. And this is sort of one of the beautiful pictures that I got from one of my experiments, and I'll tell you about it a little later. So the combination of these two proteins basically gives us not just fluorescently labeled pretty cells, but it also helps us sort of control the activity of these cells. And so this is basically the tool, the basic sort of premise of how we study memory. This is an illustration of the Chandlerodopsin protein, which has a GFP tag. You shine light on this protein, and you have ion movement that goes through this protein. It's a transmembrane protein, and you can basically control how a neuron reacts just by shining light on it. And this is an example of what we do. We try to express these proteins inside the brains and neurons of mice, which is our model system. And we use tiny little octrodes or optic fibers to sort of shine light in the groups of cells that we like or we want to study. We're sort of thinking back and forth a little bit. I'm sorry about that. But yeah, where is memory in the brain? This is sort of, again, a question that a lot of people have asked. It is in one place and across multiple places. So depending on what time you're asking, depending on how early in memory formation you're asking, and how late in memory formation you're asking, memory in the brain is sort of both localized and distributed. But hippocampus, as you can see, I've labeled both the mouse brain and the human brain, and hippocampus is sort of the key structure that is involved in memory. And sort of short-term memory and fresh memory is usually found in the hippocampus, and then long-term memory sort of this is sort of distributed throughout the brain. So a couple of things you need to know before we start about talking about my experiments. What is an engram? So I told you about Richard Seaman before, and Richard Seaman is going to come back now. An engram is something that Richard Seaman coined as a term to say that basically an engram is an imprint that a memory leaves on your brain. Now, he didn't have sort of the tools and the wherewithal we have now, but basically his sort of core concept was right. Learning something or remembering something changes your brain. And this is what he meant by imprint. And this imprint is basically what he called engram. We in our lab use fear memory. It's a very strong memory. It's very easy to learn, undoubtedly, and it's very easy to elicit. And so we basically use something called contextual fear memory, where we try to associate something in the context, in the environment of the mouse, and we link it with something very painful or very fearful. And this sort of makes like a very good tool in our hands to study how memory changes over time, either in the short term or in the long term. Natural recall, when I say natural recall, it simply means what you do every day. When you sort of sit and you try to remember what you had for breakfast today, or when you sort of sit and try to remember what you did yesterday in the evening, or like 2 days ago or 3 days ago. So it's just simple natural recall. It could be cues. So, you could see something that reminds you of something. So that external thing is called a cue, or it could just be free recall. In our human beings case, we do a lot of free recall. And light activation of engrams. So this is something I will try to get into a little more in detail. But this is basically a way of activating the same imprint that the memory leaves or the learning leaves in your brain. And this time we do it with optogenetics. So we use, like I said, channel rhodopsin and GFP, and we use light to activate these engrams. And this is called light recall. Now, what is a context? I know I said cue and context, and they're sort of hard to understand terms. In our experiments for mice, we have different types of context. Different types of sensory inputs, like, you as a human being, you see things, you hear things, you feel things, and you smell things. Of course, we also have taste, and we have interoception, we have many other things, but sort of breaking it down just to the basics for a mouse. So visual is one. This is an example of like a visual cue that we give mice inside the experiment box. Olfactory, a very strong cue for mice. In our case, we use, for example, we use many odorants, but one of the odorants we use is vinegar. So it has acetic acid, or vinegar has a very strong smell. Somatosensory, what you feel in your hands and feet. So whatever exposed parts of skin that you use to explore your environment. In our case, we sort of use different grids so that the mice can walk on them and sort of feel whether it's a square grid or a long grid, and the spacing between grids is they're very sensitive to that, and so on. And auditory. Sometimes we play pure tones, sometimes we play white noise. So these are all different kinds of cues that the mouse gets from its context. So this is really the workhorse of our lab. And I'm going to sort of try to explain the tool as is. So if I could sort of direct your attention here in the bottom part of the image, we use something called, we use a transgenic mouse. It's called a cfos-tTA mouse, and I'll tell you why. So cfos is an immediate early gene, and it sort of gets switched on and it gets expressed whenever a cell is active. In this case, neurons in the brain. So whenever a neuron fires or is active or is excited, the neuron itself expresses cfos. And now, like every gene, it needs a promoter and it needs sort of a downstream region that expresses. So what people before us and what some really intelligent scientists, neuroscientists decided to do was to take the promoter of the cfos gene and attach something called a tTA, or a TET transactivating element. So the molecular biologists in the room will know what I mean. It's sort of the tTA-TRE system is a very well-known, very well-used system. It's basically two proteins that have an affinity for each other. So what we do is you take the cfos promoter and you put a small gene for the TET transactivating element right under the cfos promoter, which means every time a neuron is active, you get cfos expression, but you also get tTA expression. Now, tTA as a protein will go bind to TRE. In general, sort of, if you don't perturb the system and you just have the system, tTA will just bind to TRE and will express whatever is downstream. And in our case, we do another small manipulation to insert the gene for channel rhodopsin EYFP, like I mentioned earlier. So we have, it's sort of a very neat, nifty way of trying to link expression of a gene, but with an activity of a cell. So this is what happens usually in a normal situation, right? So you have some sort of cellular activity, cfos expresses, tTA expresses, tTA goes binds to TRE, and you have the expression of channel redoxin EYC. Now, for us, it's important to, if you want to use it as a tool, it's really important for us to restrict spatially and temporally the expression of these genes, right? So you want to have really good control over where these genes are expressed and when these genes are expressed, because these are very specific proteins and you need them in very specific places at very specific times. And so if you come up here, what we do is we basically take the second part that I explained, the TRE channel EYC, and we package it into an AAV9 sort of virus. I know virus is a bad word nowadays, but trust me, these are not infectious and they're sort of attenuated and they're very useful tools, at least for us, when we study things like memory and behavior. So AAV9 is basically a viral coat. You put your plasmid in there that expresses TRE channel EYC. You put it in a tiny syringe, and then you just inject it where you want the virus to express. And in this case, we do it in a small area called DG, which is basically part of the hippocampus. And you do it in a transgenic mouse that has this cfos-tTA construct in it. So as you can see, you're basically combining these two tools together to sort of prep the system to say, hey, you have tTA will bind to TRE, and you'll have channel expression. Now, this is all well and good. This is spatially restricted expression, but what do you do for temporally restricted expression, which means the instant you put in the tTA and TRE, everything is just going to, the instant the cell fires, it is going to keep expressing channel EYC, which is not what you want. You want like a very time-restricted expression of these genes. And so that's when you have doxycycline. So doxycycline is an antibiotic, but it's also used to control the expression, because doxycycline in a normal case will bind to the tTA protein and not let it bind, not let sort of the whole complex bind to the TRE region. And this is very useful because in the mouse, we can literally control when we want the system to switch on by giving doxycycline in the mouse's diet. And I think it's a super powerful tool, and I've used it so many times, and it sort of never fails to amaze me. And this is sort of an example of how this works. So you see, you have HC's home cage. So you have a mouse on day one, and it has doxycycline in its food, and it's sitting in its home cage. It's just being a mouse. You put it into a box, sort of a different box on day two, and you give it a shot. But while you're doing this, you remove doxycycline just before you do this. And when you do that, basically you express channel EYC in your sort of cells of interest. And in your case, it's going to be where you injected the whole construct, but also temporally. You can say, I want to do it on day 2, I want to do it on day 1, I want to do it on day 3, I want to do it on day 10. And so it makes for a really powerful tool in terms of both space and time related expression of what you want. Right. So there are some very sort of basic conditions to call something an engram. So when I talk about excitation of cells and excitation of neurons and networks, not every cell or not every neuron that gets excited is part of an engram. You need to have persistent changes in specific neurons. So like I said before, these cells are called engram cells, but these are also sort of changes that last, right? So it's not something that happens today and it's gone tomorrow. Our experiments and behavior experiments last over days. And so they have to be really persistent changes. They should be able to be reactivated upon appropriate retrieval conditions. So for example, if you label a fear memory, you should be able to reactivate that engram to elicit the same behavior you did as before. And in our case, it's going to be freezing behavior, which is related to fear. And the activation of these engram cells, like I said, should elicit memory retrieval. So these are sort of the 3 very basic conditions for when you call a group of cells an engram or a memory engram. And this is one of my favorite videos. I love playing it in most of the talks that I give. So this is basically light-induced freezing in a fear conditioned mouse. So as you can see, the mouse by itself is totally fine. It has a little hat on its head. This is because it has had bilateral injections into its hippocampus. And it's basically, we've taught it that every time we shock it, it's scared. But as you can see, we are activating, before we already labeled this engram, but we are activating that very same fear engram, but this time not with shock, but with light. And as you can see, the mouse remembers, but like artificially remembers via light, that it's sort of fearful. It doesn't know what it's fearful of because it's not getting a shock, but basically you're activating the same fear memory. And the behavioral output here is freezing. And as you can see, the mouse is moving around a little sort of baseline movement, but in general, the mouse is sort of scared and sitting in one place and trying not to move as much, because basically we're sort of making it remember a bad thing. And this is a normal reaction to that. Now, this is actually my segue into what I'm going to talk about. I wanted to talk, so since we use fear learning and fear conditioning, the hippocampus is not the only one that's involved when you talk about fear learning and fear conditioning. And there's a beautiful little brain region called the amygdala. It's an almond-shaped nucleus. And this is really key for memory, for emotional responses like fear, anxiety, aggression. So all of these sort of very emotionally valent responses. And sort of the fun fact is that it looks small, it looks weird, it looks like it's, squeezed into one tiny part of the brain, but it has a very defined anatomy, and the anterior and posterior parts play very distinct roles. So even though you would consider anterior and posterior amygdala or basolateral amygdala as part of the same complex, the anterior cells do entirely something different, and the posterior cells do entirely something different. So for something that's small, it has like very high rate of specificity and sort of controls very specific behavior. And so I know it's a little old, but I want to talk about a classic paper that came out from our lab back in 2014. And maybe hopefully at the end of my talk, you'll understand why I chose this paper. It sort of spawned a whole way of thinking about integrating engrams and sort of understanding what memory means and how it, how to understand and what kind of tools we use to understand memory. So this is a paper that came out in 2014. It's called Bidirectional Switch of Valence associated with the hippocampal contextual memory engram. So it's a little self-explanatory, but I'm happy to give you sort of more details. I think it's a very cool paper. Mice, viruses, and hardware, just like I mentioned before, we used a very similar system. So we used the cfos-tTA transgenic mouse, like I said before. We use, so instead of TRE, chalarodoxin GFP, this time we use TRE, chalarodoxin mCherry. And so the only difference is that the same cells are marked, except they look red now. And we do this both in the hippocampus and the amygdala, but I'll tell you why the hippocampus results are really interesting. So yeah, like I said, channel mCherry into the hippocampus and amygdala, and then we do the injections, and then we implant the optic fiber immediately, just so we have access to whatever cell groups that we've labeled. And so later, when we train the mouse, when we do the fear conditioning, we can activate or inactivate the group of neurons simply by switching on or switching off the specific kind of light. Yeah, I just like showing these. They're so pretty. These are single neurons, you can see in the dentate gyrus, part of the hippocampus, and these are sort of the dendrites of the neurons. You can see this is the cannula, the place where the optic fiber comes in. So if you imagine the light is sort of more or less conical, you do get like a bunch of cells that are under light control. And this is how the amygdala looks like. And this is sort of the cannula part for the light, for basolateral amygdala. Now, we use two different kinds of behaviors for this study. Very, very sort of basic, and I'm pretty sure like the psychologists and sort of cognitive neuroscientists will understand this. So first one is called shock place avoidance. You just take a mouse and you put it in a box, sort of a longish rectangular box, and you let it run around. And this is basically its tracks for the first 3 minutes, and let's call that baseline. You find out that there's a slight increase in the left part of the box where the mouse tends to spend a little more time, and you call that the target zone. Now, the next 3 minutes, you make sure that every time the mouse gets to that left side of the box, you shock it, right? So, of course, once you do the sort of regular shocking, the mouse will not remain in that left side of the box. It will try to spend as much time in the opposite side of the box, which clearly does not want to be shocked. And this is with the shock. So then you test it again without the shock, just to be sure that it understands, and you can see it's like a much cleaner result. So basically just avoid, the left side of the box. And you do it again, just to make sure, you have the memory there. And this is basically how the analysis looks like. So you take the time spent, during you take the time spent away from the target zone during these two sessions, and then you sort of just remove the baseline, time from it, and that gives you this thing called the difference score. And so basically, the more negative the difference score, the more it goes away from the target zone. And you see why this is important, because we're going to talk about another behavior where the difference score is going to be positive. So this is shock place avoidance. This has no optogenetics whatsoever. You could just take a regular old mouse and do this, in a behavior box. What we ended up doing was we ended up replacing the shock with light. So you take an animal, you take the cfos-tTA animal with the Chandler rhodopsin and M-cherry injected, you keep it on doxycycline, and you do sort of the place avoidance. You make it sort of learn what place avoidance is. Every time you shock it, when you do sort of that shock protocol, you remove the dox. So basically what you're doing is all the cells that get excited during the coding for this place avoidance get labeled also this time now with Chandler rhodopsin and M-cherry. And then what you do is you put it back on dox, and instead of the shock this time, you use light. So you basically excite the same cells that were excited during the shock. And now basically what you can do is just replace the shock with light, and you will have the exact same behavior. And you see, we did replace the shock with light, and the difference scores look similar. So it's not as strong. If you remember, it was about -50 if you just do shock. Of course, shock is a lot more stronger, but you still see it's still quite significant. I mean, you can replace shock with light and still get the same results. So yeah, you do the same thing where instead of shock, when it goes to the target zone, bam, you give it some light and it realizes that there is something fearful there, moves to the other side of the box. Yeah, and then we were like, well, negative memories are all nice and fearful memories are easy to understand and very easy to use. But what about positive memories, right? So that's the other end of the spectrum. What would happen if we switched shock for something positive? In our cohort, we end up using male mice. And so a positive sort of cue is going to be a female mouse, right? So instead of doing shock in the target zone, place a female mouse. And as you can see clearly, the male mouse is very interested. It spends a lot of time in the target zone. And the difference score is positive. So, the score shifts as though it's going to spend a lot more time where it wants to spend because there's something positive there. Again, we being what we have, we being who we are and the tools we use, we try to replace the female with light. So you do the exact same thing. You do the place preference. You remove dox just on the day of training when you're exposing it to the female. Some cells will fire. A network of cells will fire. You label that network, and then because the network now expresses Chandler rhodopsin, you can simply shine light and stimulate the same network at a later date, even when you're on dox. So you basically just replace female cue with light. And as you can see, the difference scores are also obvious. You can literally, every time it goes to the target zone, you can sort of shine light on it and it will stay there longer because it feels like there should be something positive there, or there should be something nice there, but it doesn't understand why, but it knows that there is something nice. And so it ends up spending more time in the target zone. Now, the really, really sort of key experiment of this was what would happen if we switch the valence, right? So you take this, I'll go through this one by one. So you take this mouse that had opto place avoidance. In this case, it makes it avoid a place because you're shocking it in a corner. You take the mouse where we trained it for opto place avoidance, and every time you stimulate that engram, you give that mouse a female mouse. So you're basically trying to change the valence of the memory. Instead of shock, you're giving it a female. And while you're doing this, you're constantly activating the same network that was labeled by shock. And so basically, you're trying to change opto place avoidance to opto place preference. So you're trying to see if the mouse the brain of the mouse reorganizes itself to understand that something that was negative before is now positive. And you do the exact opposite with the other group. So you take a mouse that had place preference, opto place preference for a female, you stimulate that group of engrams, and instead you shock it. And you try to see how the behavior changes. And this is probably the reason why this paper is one of my favorites. So basically what we did it actually does switch the behavior. So you took a network of cells that coded for a memory that was aversive, and you switched its valence to now code for something positive. And so you can see on day 5 and day 9, on day 5, a mouse trained with octoplace avoidance has like a difference score that's super negative, but that negative sort of value decreases on day 9 after you switch shock for female. And that difference score that was positive for female decreases drastically when you switch female for shock. And so this is probably, like I said, it's a sort of a very landmark conclusion. And please go through the paper. I don't want to do sort of a journal club to give you all the graphs, but it's sort of very paradigm shifting, because the tool that we have, the engram sort of labeling and optogenetic tool, it helps us not just understand something in terms of what's happening in like a very small network in the brain or a very small region of the brain, in this case the hippocampus, but it's sort of massive multi-brain sort of across brain region changes that end up actually changing behavior. And that's what's sort of really important, right? I mean, a brain drives behavior, and you can changes in the brain have to be significant enough to cause changes in the behavior. And so basically the conclusions of the study are that a dominant valence, either positive or negative, that was associated with the hippocampal memory engram was actually reversed at the cellular level. So we did this because we stimulated groups of cells as part of a network, but we really did it at the cellular level in a very specific region of the brain, and in this case, in the hippocampus. We demonstrate that emotional memory is malleable, it's flexible, and that can be updated and changed over time. And the connections between the hippocampus and amygdala are quite plastic. Like I said, the amygdala is important for emotional memory, the hippocampus is important for memory as such. But we show that basically, or this study shows that basically the connections between these two regions are quite flexible, and they can change over time and with changes in environment and cues. And we also demonstrate that the cellular and network changes across brain regions, basically it's across brain regions, like I said before. These are both cellular and network level changes, and across sort of millimeters in the mouse brain. And this can sort of give out good behavioral changes, or significant behavioral changes. And so I always like to finish with asking, how is this all applicable? Because our work is very basic and sort of very, very basic neuroscience, and gives very basic insights into how things work. I'll just sort of mention a couple of papers that came out around the same time and right after from our lab. One of the things we used memory engrams to study was also to look at amnesia. So amnesia and retrograde amnesia is really an issue. We don't know what amnesia means, and I would highly encourage you to read this paper. Basically, it talks about how protein synthesis is really important, and if you block protein synthesis, you block how memory is encoded, and you block the whole formation of the memory engram network. This is, again, another very cool paper where you look at, basically you use the learnings from these two papers, and you look at what happens in early Alzheimer's disease. And it was led by a really sort of favorite colleague of mine, Dheeraj, who was a PhD student in our lab. He basically saw, I mean, the sort of the crux of this paper is that memories in an Alzheimer's mouse are not lost. So the engrams of the memory still exist. What actually happens is you lose access to them. It's a very elegant piece of study. I really recommend that you go through it. And then finally, this is one of the more sort of recent papers that came out of our lab. This is more related sort of to more hippocampus and amygdala, and we look at what sort of the dynamics of fear memory and fear extinction memory. And extinction is basically sort of forcible updation or forcible forgetting of a fear memory to something else. And this is more or less my sort of area of what I study. And Shangyu was a PhD student in the lab who sort of did some amazing work, used engrams and a bunch of other tools like calcium imaging to understand what exactly happens sort of in the amygdala over time as a fear memory sort of fades away. Does it fade away or does it come back? And sort of all these dynamics of things. And I end with that. So the main paper I talked today, these are the main two authors, Roger and Steve. Roger is currently at Rush, and Steve Ramirez is at Boston University. He's an assistant professor. And that's our lab right there. Thank you very much, and I'm happy to take questions.